So I'm also going to record this one as kind of a preview, but a lot of the strengths of these sessions are if you can actually be kind of exchanging info and working together a little bit. Don't worry, I'm not going to make you do the breakout rooms ever again. That was a disaster. Zoom never auto-completes how I want it to on email, so don't worry. So in this case, and this is a real life example right here. This is something I had to figure out. There are plenty of drug inhibitors that don't just wholesale kill tumor cells. Some of them just stop them. Why would this be, number one, if you're into medicine, why would this be a better thing sometimes? Not only if you're into medicine, maybe if you're also into ecology evolution a little bit too. So go ahead and think about this. We will kind of work it out together a little bit.
Wait, can you repeat the question one more time? Like, are we asking if it's what's beneficial for them to stop, like just to be frozen?
Yeah, that's more, well, that's more like green text, yeah, for thought, like food for thought. Why is it better sometimes? Oh, let's see, is this gonna hook up here? Probably not, so you guys probably can't hear that, shit. I just realized everybody in the room can't hear you guys either. I'll just repeat the questions each time. So in this case, the question is, this is kind of getting at the food for thought idea behind cytotoxic versus cytostatic. If you kill everything in a tumor, what does that leave room for? Versus if you just freeze things up. Now this is more an application because this isn't never going to be on an exam. I'm never going to ask you gigantic cancer biology, philosophy questions of how to treat. But it doesn't hurt to see that that question's being asked. So in this case, we have our cell cycle and we've been going over that a little bit. And as you can tell, I'm not really focusing on the mitosis sections right now. Always try and separate those a little bit. A lot of the key parts of the cell cycle, they're at these checkpoints. They're less so on that decision to undergo mitosis. It's an important decision, but it kind of just goes once it's told to go. It doesn't have as much thought behind it as some of the checkpoints do. So after this big one, we'll kind of open it up to some Q&A as well, and I'll post the other question while I'll answer some Q&A while that other question goes up. But for now, if we're looking at mitosis, and in this case, so let's say there's our M. Oh yeah, here we go. So here's our mitosis phase. You can go into G1. Well, this. Oh shoot.
Oh, now Zoom's updating.
Oh.
You want to allow your device to make changes. Yeah, for choice.
Here's our basic look at the cell cycle. Is there a molecule that's changing in this time, and is there a cell structure that gives away what section these cells are stopped at? All right, so the big molecule of all changes during this cycle. What is the big molecule that changes in this cycle? What's that?
Well, like the DNA? Or like, are we talking about the go and stop signals?
Sorry, yeah, they're coming in here. I'm gonna make a way to work this next time, sorry. So in this case, somebody said DNA and somebody said cell structure, right? 80K. Oh, like the CDKs? Yeah. That's good, yeah. That actually counts. That's not what I was expecting. All right. Yeah, this is good, and this is why. So we have multiple right answers. Number one, DNA. DNA is gonna go from ×1 over here to ×2, remember? because of synthesis, or because of, yeah, synthesis phase. This is some evidence here. Another way is that as you progress to this stage, you get more cyclins and CDK molecules. This is a good catch. I didn't have that on there. That'd be a right answer no matter what. Good job. My main thing that I was actually just looking for was, yeah, where's DNA at? So anything past here in this section, if the DNA is doubled, you know that they're frozen post S phase. They have to be frozen in G2, or they have to be frozen right during mitosis. So what's something that you could use to differentiate between being frozen at either of these cells right here, either of these stages? Remember the final, oh yeah, remember the final checkpoint. What is that named after? Growth? Yeah, so at this stage, they're done growing. But if they're stuck at this stage versus this stage, what's the one thing that separates a cell that's in mitosis versus one that it's grown? And you're right, but it isn't splitting yet. Spindle, yep. So that's right. So we have a spindle. So if you had to designate between G2, that's not a very good two, G2 and mitosis, only mitosis has the presence of a spindle. So again, in terms of massive cell biology, I'm definitely not going to assess like, hey, which microtubules are pulling, pushing, things like that. But it's not a bad thing to know their existence, right? Especially for big stuff like this. Awesome. Okay. Now, yeah, and so we'll kind of go through things at this pace, and like you never have to like shout out stuff. You can if you want. If that helps jumping in, asking me questions, that's good. We'll go there. Okay. Yeah, and so this was awesome because we actually talked about as CDKs / cyclins rise. That was a very good addition. Right. There's multiple things that can kind of leave kind of a bread trail, kind of a crumb trail as far as what's happening and what stage you're at in mitosis. And it's not always that, it's not always the same stage that gets deregulated first. It's not always the same checkpoint that gets deregulated first. That can change on the type of cancer or disease that you're looking at. Okay, so that's kind of how I'll intro these. Just sort of like, here's a big one. This will never be on the exam, but ultimately it's a pretty good expression of, it's a pretty good expression of the material. And I'll always try and kind of make it a little more advanced since we'll be able to work through it together. So in that vein, is there any Q&A that I could maybe just draw out? So I'll kind of have these pauses once in a while. Yep. So the CDK phosphorylase cyclin? Yeah, so they kind of work in a partnership together, yeah. So one of them is, I believe CDKs are there dormant. So the question was, what happens first, CDKs and cyclins? And CDKs are there dormant and they're inactive, I believe. The cyclins start showing up when the cell cycle starts to inspire itself to go. Like at G1, it's like, okay, like let's rev some of these up and they start to come together. So in this case, I believe you just have some CDKs laying around. They can't do anything without their cyclins binding to them. So then once the cyclin shows up, you have your go signal. Now the cyclin signals come from the checkpoints of G1, and these cyclins are only going to be inspired to show up if you've got enough food, resources, things like that to grow. Because remember G1, that's mainly concerned with, can we make another cell? Do we have enough lipids? Do we have enough DNA? Do we have enough, carbs, proteins, things like that? So from this point, the superstructure is going to go on and add phosphorylation groups to all kinds of stuff and all kinds of go signals. And this is the ultimate trigger of go. Well, it releases the ultimate trigger of go, which is that MPF molecule. Once you trigger enough CDKs and enough cyclins together, and you get MPF out and ready to go, that's the end of the game. Mitosis is gonna happen unless there's something really wrong with the cell. Yep. Yeah. There's a bunch of things that actually are gonna inhibit these two things right here. So let's actually meet a couple. Okay. Cyclins. So I can't remember all the stop signals that are outlined there. Anytime I ask you an exam thing, and this is another good version for these meetings, is I can be really frank about what's on an exam and kind of guide you through that language. I'd always make sure to tell you what something is doing if I ask you about it. So in that context, let's look at something, a gene called P53. P53 is your ultimate tumor suppressor. It's bigger and better than BRCA1 and 2. What it does is it runs around and it basically blocks this formation and it stops your go signals in their track. It's very careful about when it does this. Okay. Okay. Yep, yeah. 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 Right. So it basically decides if it's going to let them go. And so P53 is going to come and get angry if there's DNA damage or if something is wrong with the cell. And it's the ultimate stopper of your go signals. This is why the worst cancers in pretty much any tissue, if they've lost P53, you're not looking at a good prognosis. Because if you've lost this, you've lost your best way to make sure that something can stop this cell from surviving and growing even more.
So basically, they decide whether the tumor is cancerous or not, because if it's benign or malignant, it'll let it continue to grow because it's not cancerous.
Yes, so the question was, let me make sure I repeat it right. Basically, losing P53 can lead to a tumor, right? Because it's such a grave imbalance of a stop signal, yeah. And that is what happens in a lot of cases. So a good comparison to show you its power is if you're born with only one working version of BRCA, for example, which is a really, really good tumor suppressor, you will go on and live a life most of the time without developing a tumor. You are at an increased disposition for one, but it is not genuinely gonna happen each time.
No, get down. Get down.
You've got this, yes, yeah. So map out somebody's personal tumor and find out which specific mutations they have, absolutely. And we can combine patients with 1,000 other patients and get like a big consensus. And associated with stop signals. Okay. It's dead. Sorry, so looks like we lost the audio on their side. Okay. Okay, how's that in Zoom world? Are you okay? I might have to talk a little close to the computer and look kind of boomery. Sorry.
Better.
Better, can't hear. Sorry. Okay, yeah. These may not be charged all the way for today. Okay. All right, yeah. So good thing is in the context of stop and go signals, if you lose one, it's not game over. But if you lose multiple ones and that imbalance starts happening, we're on the road to making that first, second mutation that can lead to a group of cells that don't behave and don't react to your immune system trying to kill them, or the apoptosis pathway. All right. So all right, so I can hear. Can you guys hear me now? Okay. Yeah, it's like replaying up in here. So I think I'll just do the extra mile and connect to the actual room next time. Sorry about that. I thought I could pull off the Zoom light. Okay, so it looks like we're back. Go ahead. All right, so Sophia's one of our student leaders. Listen to her. She's taken this class and done really well before, so not too bad of a model to follow. Looks like this is working pretty well.
Okay, so my name's Sophia, and let's look at this problem that deals with meiosis. So you have 3 different drugs to choose from. It looks like drug A causes DNA mutations, drug B causes the spindle attachments to fail during meiosis, and drug C causes some chromosomes to replicate themselves a few extra times. And if your aim is to induce a new polyploidy, is that how you say that?
Yeah.
So pretty much taking the whole genome from a plant, and it looks like we're doubling it. Which drug should you use to accelerate this process? Does anyone have any thoughts on this at all?
Which one's definitely not going to work?
Yeah, which one's, yeah.
Wait, what?
Wait.
No, it's okay.
It's okay, why do you think
It's not
B?
an exam. Yeah, this is good.
Why do you think B?
Somebody was saying B that will work, I think. So you're actually on the right track there. So I'll just say it over here. So an answer was, why not B? Because if the spindle doesn't pull, it's going to have an empty cell, right? But what about the side that it did pull to?
It'll have.
Mind blown. Mind blown, yeah.
It looks like someone
That sounds
said
good.
drug C. Are you saying drug C will or will not work? Jessica. Will work. Okay, so she thinks drug C will work here. So drug C is the one that some chromosomes replicate a little bit too much. Yes and no, because you'll just have an extra amount of cells, kind of like when you won't go up in that ploidy, or however you say that word. You won't go from 2N to 4N. You would still be at 2N, but you would just have an extra amount of cells. You won't have an extra amount of cells in the same cell, if that makes sense. Exactly. Yeah.
Right, there we go.
So I don't know if you guys could hear that, but he said, was it an example you used in class?
A little bit, yeah.
How when you pull those chromosomes all to one side of the cell, it'll up the number in chromosomes, which leads to that raising the ploidy number. Yep, non-disjunction.
Right. So here's the details behind drug A. Drug A would come in and we'd make a few nicks here. We'd have a couple new things, but we'd have no chromosomes. We'd have no gains in this case. We'd have new mutations, and there's things you use that for. Drug C. If suddenly now I have like some region here, an extra region of this part, I have a region of this arm, and maybe a little region of this arm over here. That's good, but I didn't make it all the way to 4N. What I need is something that's going to induce a new change that's going to copy the entire genome that I can engineer later. That means that I want to interfere with the spindle. The spindle is over here, it's going to attach. Same thing over here. Under normal circumstances, you get one over here, one over here, one over here, and one over here. This is just a regular single N in this case. We don't want that. What we can do with drug B is ruin the spindle, and let's say eventually cause a cell to bring both copies over here and induce a non-disjunction event. And what we've done basically times 2 our genetic material. Ooh, can't tell if I misspelled that, right?
Yes, it is drug B.
Right, yep. And
Yeah.
so this is the very beginning of a new species that has an entire extra copy of the chromosome that it's going to pass on when it meets another full chromosome.
Offspring would then have that, and then they would have that, and they would have that. So
Right. Yep. So by inducing it, oh yeah, so I'll answer this. All right, so basically you're ruining the spindle on one side. Yes, yeah. So if we send in a drug that's going to ruin the spindles, eventually we're going to get a haploid that we ruined it on one side, induced a mega non-disjunction, and the whole thing came over with a whole new set of genome that's going to get treated like it's fresh DNA.
Yeah, and something I kind of want to add here is don't think of non-disjunction as it always is going to be doubling, because there will be some times where it can just, like he said, with pulling both of them over, can maybe will only ruin the spindle of one, and then you'll get a certain amount of chromosomes there. So just think of non-disjunction as pretty much the cell is not following the correct pathway to that ploidy that you want.
So luckily, yeah, so that was a good question. So the question was, once you induce this polyploidy, is that going to ruin the cell? Sometimes, yeah, and that's the beauty of GMO organisms. We're not being nice to them eventually, but if a plant does work in this case that has more chromosomes and more genes and more DNA, you produce more product. And that's kind of the idea we did with strawberries. We didn't really manipulate them too much. They started out 2N and now they're 8N. They have 8 copies of each chromosome.
But yeah, definitely in humans, you can see different syndromes that come from it.
Yeah, so the more
Some
complex,
being more
yeah,
lethal and some not.
for sure.
Is it worse to have it initial or later on in the division?
Right. So is it worse to have a non-disjunction event happen at meiosis 1 at that first split, or at meiosis 2 at the later split? That's a good thought question, actually. So give that a sec and think, which one is worse? Actually,
Okay.
oh yeah, we just muted. Don't worry, we're here.
No, it was just muted.
Sorry. Okay. So I'm going to actually speed down to down here and do this right here for non-disjunction. So imagine these are a maternal and a paternal version of a chromosome. Highlighted in, oh, well, those look like cells, so let's just do that. Split 1 versus split number 2. So if you have split number 2 being the event, that means right now everything's okay. You actually have sister chromatids of each of these, sorry. Okay. So in this case, if we're splitting at the second division of meiosis, or if we're having the non-disjunction event occur here, and let's say it's going to occur in this cell like we saw in class, half of your haploids in this case of this division are going to be normal. If the pull happens over here, you're going to have one where you're missing one, and you're going to have one where you've got too many. So all said and done, we have two basically atypical cells that we're passing on in this case. Now, if you split over here and you bring the sister chromatids with each other, and you start with an empty, automatically, you've got one that's missing, you've got one that's missing. And now you've got two that are 2x. So the little H's that I'm drawing, sorry about that, that's sister chromatids being connected. Because remember, usually they're going to split into this nice single piece, right? Single chromosome.
Are
Right.
these haploid cells then, or meant to be haploid cells at the end?
Yeah, so we want everything to be haploid. We want just one copy of each chromosome showing up.
What happens if the cells that have no DNA bind with the cell that has double the amount that it's supposed to? Would you then get like a normal offspring, or would that just be a clone?
No, so in this case, you would only have the contribution from the other parent in that case for the fertilization. So let's say we're talking right here about chromosome 20 this whole time. That's what we're visualizing, let's say. In this case, if you have offspring down here in cyan down here, you're only going to have one chromosome 20 if this is what fertilizes. You're only going to have one chromosome 20 if this is the one that fertilizes, assuming the other parent has a normal setup.
I mean, like, what if it was like where the one where you have two, like you have double?
Oh, yeah, let's get to that. Yeah. So you're going to have 3 in that case because you're bringing 2 from here plus the 1 from the typical parent.
So for the gametes that are monosomy, would they just be one N after the next the next, you know what I'm saying?
No, help.
Like after the next division, would they turn one N whereas they're 0 right now?
So are you talking about this one right here?
The very end, like where it's the gametes, where one is monosomy. And you said
Oh,
that's
right.
from one half of the gametes, correct?
Oh, you mean in cyan right here?
Yeah.
Yeah, so that's the total that the full-on offspring is going to have. And you want to.
Oh, so that's the full on. That's from both
Yep.
parents, both divisions.
Yeah.
Okay.
Yep. So in context to the question, is it better to have a split at the first division or the second division? If you split at the second division, you've at least got 2 out of 4 that are normal. If you split at the first division, you've got none that are going to be your typical passing on chromosome structures. Now, I think this is a good point that I should have brought more in class. Individuals that are missing genetic info can lead pretty normal lives. Okay, that shouldn't be always injected like the language we saw with the legal precedents that they were trying to make in that. This is a good example where genetics aren't fate every time, which is something that I do want to impress on everyone. Cool. Okay. Well, dang, we're getting our money's worth on this one. This isn't bad so far. I was worried it'd be like silent or something. That'd be worse. Hey, my time is accurate now on the iPad. I fixed it. Yeah, as you can tell. That always made for a good like dad joke each time. Like, oh, you're stuck here, haha. Now you are actually though. It'd be terrible. So then there's always time for stuff like this. These are kind of reflective of those guiding questions. This is actually something we haven't actually got to this on Thursday. This first one, I'd say. We will go over this, basically, why are recessive genes recessive? In that case, the short answer is that a dominant allele is a functional protein, and it will go and do something. A recessive allele is usually whatever DNA change is there, kind of messed it up. That function is pretty much gone, so it's kind of a blank slate. That's why you only need one dominant to show the effect. Now, not everything's Mendelian, and not everything follows the strict dominant versus recessive rules. So this is actually well timed because this is kind of the halfway point. Yep. All right. Pretty good. So kind of general stuff too. I've been talking a lot. Does anybody have something specific? Things like that. Yeah, that's a good question. So the question was the cloning all blue text? I would say I would probably give you any procedural stuff like that in the question. And I'd probably say, remember, when you're cloning, I'm going to take out the nucleus and put in a new nucleus with DNA. Does that have the complete setup? Does this, yeah, it would mainly be in a context that would be addressed, maybe addressing something different in that case. But no, that's a good question, yeah, because there wasn't any title on that one and I spent some time on it, right? It shows 2 chromosomes. Yeah. Yeah, so the reason that it shows 2 chromosomes and it's going to become trisomy is that you're getting one from the parent, from the other parent in this case. Okay. But yeah, in the context of cloning, the principle itself is pretty simple, right? Like if you put in DNA into a cell, you put it in the right environment, it will likely act like that cell. Beyond that, I mean, I'll probably use that to assess mitosis, meiosis maybe a little bit, but I'd always provide that context because that was blue text, I'd say. That's why you're writing about it. Monosomy, okay. Okay, so that's where the monosomy comes from, the other ones that don't receive any, exactly. So if you're getting 1+ right here,
and 0 right here, here's your grand total for the offspring. Yep, so you're good. Yep. Oh, same thing, yeah. Yeah, this is, yeah, so this is the possibility. So this is if the split occurs, no, let's do this. This is if the split occurs here. This is if the split occurs right here. And it's uneven and it draws over one of those chromosomes when it shouldn't. And yeah, Callie, that is what we would, for on a genetic instruction standpoint, you're looking at a human for 2 copies of each chromosome. So I would say that if you lose or gain one of the big chromosomes, so the size of a chromosome goes up from chromosome 1 or goes down from chromosome 1 down the line. So chromosome 17 is pretty small, chromosome 2 is huge. If an embryo is born without chromosome 2, that embryo will likely just never take. It's missing too much. There's thousands and thousands of genes on chromosome 2, not enough. Similarly, if you had a 3rd chromosome 2, like one of the big ones, it's an overdose of instructions for the embryo. And this isn't development, luckily, because that's a hard class. But if you have too many instructions at the wrong time, the embryo, again, is going to fail. And medically, the failure won't happen. The failure will happen very early. It will not take, if that's the right word in that case. Okay. Whew, this is fun, kind of. Not for you guys, because you have to get tested, but other than that. No, yeah, so I mean, they'll get fertilized. So the question was, will the empty gametes in this case still fertilize? In the context of this question right here, we're just looking at 1 chromosome. Yeah, so if this was fully empty and you just got 1 N from 1 parent, ooh, yeah, I don't think anything's gonna happen then. It's just not enough instructions. Yeah, they'd probably fuse and nothing would happen. It's just not enough instructions. That's not always the case with less complex organisms. So sometimes you can induce that and cause a lower amount to function. So like we saw with banana, the 1N banana had to come from somewhere. Okay, are there 2 divisions or 1? There are 2 divisions in meiosis. 1, we'll do number 1 and number 2. This is why you end up with 4 cell products at the end instead of 2, like in mitosis. Yes, yeah, so at the end, the numbers in cyan are what comes after the parent. The other parent donates one of their chromosomes. So 1N plus 1N is going to equal 2 under most circumstances. It's only when you have all this craziness that you start inducing differences in chromosome number. Dang, I'm behind in the chat. There we go. Cell chromatids paired with the 1, they'd be fine. So the cell with extra chromatids, sorry, Elijah, I'll get your question. So if the cell with extra chromatids paired with the 1 with none. Oh, wow. Yeah. All right. So I don't know if everybody can see Elijah's question, but it's good. You stole one of my exam questions. So if a cell with no chromatids of, let's say, the exact chromosome that we're looking at, C20, if you end up with a cell, I'm going to go do a fresh one for this. If by some crazy, ungodly chance, you end up with a 0 haploid for chromosome 20, and parent number 2 comes in with a double accidentally of chromosome 20, you're going to be fine. Oh, yeah, sorry. New messages. So in this incredibly, incredibly rare event, we're good. The individual here would be much more, let's say this is the paternal side, it would be much more a contribution of this chromosome in particular of the father in this case than they would be the mother. This would be an atypical relation like passing down, but you have to have lightning strike here and have a 0, and here at the same time and have 2, and they have to meet those 2 out of the millions. So you're telling me there's a chance. It's possible. I'm glad you brought that up. I saw this on an exam on undergrad, and it was really hard. Took me a little bit. Yes, yeah. So I mean, in this really atypical case, I gotta promise you, like, I think there's probably case studies of something like this, I'm sure, but in this case, yeah, 100% of both of their chromosome 20s are from the father in that case. So in this case, you'd have an offspring that is not 50-50 like its parents, it's like 56 like its dad, maybe. 2 sister chromatids, what would happen? All right, Lindsay, I'll read yours. Okay, so if the parent only had 1 chromosome instead of 2 sister chromatids, what would happen to the offspring? So in this case, Lindsay, if that happened, we would just have kind of the normal scenario that we outlined up here. If you have a mother contributing an empty chromosome 20 because it got pulled to the other side, typically the paternal is going to have just 1 chromosome right here. And so this offspring would only have 1 version of chromosome 20, just the one from dad. So that's actually a pretty good math example of if somebody does have a trisomy of a certain chromosome, they're technically a little bit more related to that parent. So it's actually a good thought. Okay. Cool stuff. Okay. Phew. All right. This is just kind of for fun. I did the one with genomes and what is it? Genomes were like the world, chromosomes are like countries, genes are like cities or events or landmarks, and DNA nucleotides, the little units that make each gene, are small pieces of stuff that, you know, some if you take away, the city goes down, others if you change or take away, city's fine. This is actually talking with Leah, yeah, so give her some credit too. This is a good example.
Genome could be like food.
Yeah.
And then I down some chromosomes, that could be like ice cream.
Oh yeah, so like all food and possible.
Or just dairy products.
Okay, yeah, so we'll do all, yeah.
And then further down the genes, let's say it's ice cream.
Okay.
And alleles will be the flavor.
That's pretty good, yeah, here. So we'll do genomes down to DNA. All right, so Nick had a good example. So this is, of all the dairy products, one of them is ice cream. So in this case, of ice cream, what genes create ice cream? There could be a specific flavor gene, or there could be a specific like flavor gene that amongst all ice creams is one unit that makes ice cream. So I'm gonna put the top flavor of ice cream ever, cotton candy, which at its base component is made of, I don't know, some awful flavoring, not a natural one. Here, I don't have enough room. Awful synthetic flavor chemical. So the more that you create these analogies on your own, not just for something like this, but anything that we see in this class, the more it can get you out of that textbook and more attached to whatever it is, and not attached like you're in love with the material. I don't expect anybody to be, but the human mind, like I've said, is very good at connecting 10 things to 10 things that it knows rather than learn 100 new things right now. And that's kind of the reason that I try to teach how I do with examples, is that sometimes those examples hit a chord in somebody and it makes sense. And on the other side of it, I think it's just good to see things and see cool examples from things. So yeah, pretty good little subdivision here we've got. Awesome. Toss in some chocolate chips with it and you've got a real ice cream then. Just a monstrosity, yeah. What is it, like brownie bits, marshmallows, cotton candy, just awful. Okay. I do actually like cotton candy ice cream with crap in it too, yeah. So not, okay, I was definitely not sarcastic. It's as real and terrible as you imagine, yeah, sorry. It's okay.
Oh yeah, so calories is grocery store, dairy, to milk to like lactose, yeah, for example. So there are milks, for example, that can change lactose, for example, so there can be different milk genes. There's different types of milk, for example, that the components can change. There's small differences. There's 1%, 2%. There's almond milk, even though that's not real milk. There's a couple other things. So yeah, it's kind of cool. Yeah, Elijah, that's a good one too. Building to the floor to level. I actually like that because it's kind of the structure thing in my head. That makes sense. And then at that level, there's an office, and in that office, there's a person. And maybe even the person, you could even go beyond that. The person's one part of that office, let's say, in that room. But so is the computer, so is some of their papers, things like that. And again, when you think of genes, remember that if you change or manipulate something about them, sometimes that doesn't really do much to them. You can have changes in genes, sometimes it doesn't really affect much. You can have changes in genes that it destroys the whole thing or it makes it super amplified too. So all kinds of
You
crazy
could
stuff.
also do the genome as a book, and then chromosome as a chapter, and then gene as like a sentence, and then DNA as like a word or something like that too.
Yeah, for sure. I think one of my favorite ways, again, like Audrey said, is to, when you're reading, like it is kind of like a book. I mean, the A's, T's, and C's, they don't sound out very nicely, unfortunately, but they are telling a bit of a story, and we'll see that with transcription translation. And you'll see, and you can imagine if you're reading a sentence and everything's going fine, and you pluck one letter out and the whole thing backs up a space, sometimes that can lead to some issues. Or if you insert a word, delete a word, the story can change. Maybe it doesn't end the story, but it can definitely change. So pretty cool stuff. Okay. All right, look at this. The reward, exam problems. This is recorded anyway, so everybody's gonna get these. But the reason I like showing these is that it can kind of diagnose how I do an exam. Nobody's ever had me for genetics. Kind of a risk reward here, right? Part of these sessions is to show you, here's what I'm kind of bringing to an exam. In most cases, they're going to be small or longer clinical style multiple choice. This is how I was trained at the Med Center. And I'm always working this out. And in a perfect world, obviously, I'd have you create some thesis, but 90 thesis is a lot. So let's just rely on the exams for now. The most typical style of exam question you'll see from me or any other health professional type: what is not an example? What is not a component? What is not a stage? What is not a helpful solution? Meaning you'll have to find 3 good ones, or at the same time, find the bad one. Yeah. So everybody's looking at number 1. Always remember with these style 2, don't take the sentence for granted. Don't try and speed through. We will only have a certain amount of time for exams. That's why it's still going to pay, despite being open note. It's still going to pay to know quick. In this case, that was an issue. People didn't believe it because blending was a thing. There was no way. We had no idea how variation popped up. Yeah, that's true. And big one, we'd never seen anything that actually passed to parents or to offspring from parents. That leaves one last thing, which usually I'll toss it in as a big contradiction. The hardest one of these are where the answer is something that technically somewhere in the class is the right thing, but it's not related at all to what I just asked. Part of that's application in that case. So I'm rarely going to ask you what's a sister chromatid. I probably provide that information before I ask you a question. So is everybody's focused on the next one, I think? All right, here we go. Not so fun when it's not humans anymore, right? 4N genomes, 32 chromosomes total. So think about, draw it out. This is a good chance to draw this out right here. You start with 32, and that's typical. There's some other information in here too. This is also a very good chance to decide which pieces of information are more important than the others. maybe even completely erroneous. Let's see what's here. Okay. Cool. So drawing it out would help, but 32 is a lot. Now this organism is 4n as it exists. That means that, does this mean ultimately that any haploids have to be 2n that combine with another 2n haploid? So how much is 2n in this organism total? Because only 2 gametes can ever meet. So if you need to get to 32, how many raw numbers do you need? There we go. So, plenty of crazy stuff drawing a 4n, right? Scary, multi-polyploidy stuff. But at the end of the day, if you have an organism that needs 32 chromosomes and it splits its genetic material in half, 2 things meet, make 32. What's half of 32? 16. So to form, yeah, so to form one diploid, so to form, well, remember it's not a diploid though, it's a tetraploid. But do you need that? Do you even need that info to solve the question, right? If an organism has to exist with 32 chromosomes, it's got to get 1/2 from someone and 1/2 from the other. You can't have 4 sources piling into the same zygote. You only have one fertilization, you only have 2 fertilization means to get to 1 zygote. And if you only got 2 and you need to get to 32, yeah, but you wanted all the clinical information. You asked for this on the board, right? That's the point. No, I know. Right. But this is meant to mimic clinical science and regular old science. You're going to have a lot of information at your fingertips, right? We have the entire Wikipedia in our phone. We have everything we need to solve everything, right? Which pieces of information are more important? This is especially true in clinical science. This is a miniature of what you'll see, let's say in a health, PA, allied health, in any sort of case. They'll throw an entire paragraph in here about here's everything that happened to the patient. What are the 2 pieces of information that you're going to need to put this symptom list together, right? So this is a miniature version of that. And I promise you, it's not here to mess with you. It's here to make sure that, you can see the trees past the forest. Is that the expression? I think I'm butchering it. Either way, important information is more valuable than, and finding that important information is almost more valuable than knowing lots of information. It's much more valuable to be able to seize on something. Now, again, this is kind of a hard question. One of the other cool things about these sessions is, hey, I might use one of these, who knows, word for word, right? It's a reward for coming and doing stuff, right? You have a question, Isaac? Uh-oh. Oh, shoot, what did I do? Autosomal? Oh, normal, non-sex nuclei. So in this case, normal is 4n though, still, for this organism, and just in this case, yeah. Cool, okay. Yeah, because ultimately, well, I'm not going to show you the non-disjunction drawings because that might mess everything up. Uh-oh. Be careful. It's my fault if anything's happened. Okay. All right. That might be actually where we're done. We're done. Hooray. And these sessions also have the student leaders kind of share their advice for what works, what doesn't work. Kind of our collection today was, hey, group together, it's really good. because sessions like that where you're like, hey, I'm confident in this. This is what happens here. It's really quick back and forth. It's really quick. Like, oh, I'm going to adjust that. That's right. And you can start crossing off what's right and what you still need to work on really quickly in a group. When it's just you and you're watching the recording or you're looking at the guiding questions, you can get lulled into a false sense of like, oh yeah, that makes sense. Only when you got a blank sheet in front of you and you can rip everything down on that should you start feeling a little more confident. And that's not to say you should memorize everything, but you should pull associations from one piece that you do know and try and branch that out into other related pieces. So yeah, reach out to your little study groups that I made, other random. They're there to help if you want, but again, you're in control of your class and everything else. So I will open this up a little bit for general Q&A. Yeah, you could. If I was in the Snapchat, I would send you pictures of Scout. I would spam it. But yeah, for real though, like if you all do want to do snaps, I know that my physiology lab groups, a lot of them, that's how they communicate. So that's good. Yeah, you can probably use the D2L to post like, hey, here's our group. So
Dr. Harder, I have a question.
yeah.
So back to the second like exam-like question.
Yep.
Did you multiply by 2 because we wanted a 2n at the end so that when you add the 2 n's, you get a 4n?
Right, so your haploids in this case are going to be 2n. Human haploids are 1n.
Okay, right.
Yeah,
so the reason that we figured it out was because we just divided 32 by 2.
Yep.
Okay.
Because no matter what, if you got to have an organism that needs those 32 chromosomes, you just got to have half come from mom, half come from dad, and that's it. Hooray. Here's our little mitosis with this thing. All right, pretty cool. So yeah. Yep. That's okay. Yeah. Yes. Came straight from my brain, so I'm slow and I'm making more, but this is how, again, this is the perfect application of the clinical question, right? Like it could be more, but it's like this is a question that I have to deal with cancer drugs, right? Like I have cells in the incubator. I have 1,000 different things to choose from. If I have an end goal, how do I get there? This is a good way to prove that concept, especially for what we're doing in class. Yeah, I mean, these take time to write, and I'm definitely trying with some practice questions, and any questions that I don't put on the actual exam, I usually put in practice. And I'll usually note any practice exam questions that are huge like this, like, hey, like this isn't actually going to be on the exam, but do try and solve this, for example, because that's going to help you. Not this question. The book is good if you're looking to go greater detail. Like if it helps you to understand exactly how the metaphase plate aligns, the book's a great spot. I'm never going to test out of the book because I think that's asking a little too much of you all. But if you can find, you should be able to use the table of contents and find the stuff that we're looking at to find the detail you need. Hopefully that's good. But again, textbooks and everything, it's another good question of, you know, it's why somebody doesn't graduate with a medical degree by just reading the books. It's all there. It's a matter of like finding the pieces that you need to put the big picture together. And so hopefully the exams reflect that competency that you can be able to do that. Lecture material. Yes, Lindsay. So everything that's fair game on the exam must come from lecture. Now, sometimes I may post like a recording of me and it's a little extra, like to, you know, mess with you guys if I haven't gotten to everything. But I would note that, but pretty much everything you're going to need for the exam. Wait, check that because I know it's strict interpretation. Everything that you need for the exam will come from lecture. And that's kind of a promise that most clinical educators make too. That is because you're dealing with a lot of dense books, things like that, that you don't want to assign like, hey, anything in the book's fair game. No, it's only lecture, so you're good. Yep. You're all set, Sophia? Okay. Okay, thank you. All right. You're good, yeah.
I know that they
Ooh, yeah, big differences off the top of my head is that meiosis is going to produce 4 cells. Those cells are all going to have half a genome, or well, sorry, yeah, a 1N genome, and they're all going to be variable and unique. Mitosis is 2 cells, 2N, and copies, clones. Humans do mitosis in every cell that we're made of, except the German cells that can pass on. Only meiosis is only happening in the sex cells of humans. Yeah. So if crossing over happens in a mitosis cell, that's where you get some bad mutations. So that'll come later in the advanced mutation section. So we're previewing that. All right. So I think that's good. Awesome. Good first one. Diploidy again. Okay. Yeah. Thanks so much.
Definitely went into way more detail than Tom did from him. And I was like, we never learned that. I was like,
we have different strengths for sure. Like I think
yeah, I think he's just he's been here for so long that he has what he wants to do down to like a science, and he's not going to go ahead and add any more material and like with
which
new
is
things.
good. I mean, he's very good at the linkage side. Like, he's way better than I will have like 1 thing on linkage, 1.5, but I'm not very good at it. I don't like it, so I'm scared. It's mainly for me like
You
kind of
took it with
conceptual.
Toma? Yeah.
Yeah.
I think it was more like textbook.
Yeah, he was
Right.
more like out of the textbook, like, and if you don't understand it, go to the textbook and like further discuss it. Yeah, but like he did not go into like I didn't know anything about what was it CR? Yeah, CR or like
and what is that? Like, I haven't heard of it.
I was like we never heard about that like being made and being or like the P53. We never heard anything about that or. So I'm glad to know it. So yeah.
It's what we call it, yeah. So like kind of in like have you seen the PDFs? Like red text is like okay, like yes, like this is real. Blue, it's a great example, but it's pretty detailed. And then green is like this is fun. That even the P53 stuff is pretty blue text. Like this will help you understand that there are stop signals. Here's one, but I'm not going to say like what is the molecular structure of P53. That'd be pretty awful. Yeah,
and
that's
so you said on your PDFs, which color is the?
Red's the big one. Red's the
Red's
one
big.
that's like this is completely possibly on the exam.
Okay,
Yeah.
sounds good. I'll at least know what to look for.
Okay, yeah. Sounds good. Thanks so much for coming too.
Of
Yeah.
course,
And,
yeah,
and
of
again, like I
course.
said, it's always like if your schedule is busy or anything, don't worry. But
Okay,
we're
so
good.
Sophia, Leah. Okay.
you can definitely email.
Yeah, okay, I was gonna
Anything,
ask
yeah, like
you.
if you have any questions that like maybe his description isn't helping you understand, because I know some, no offense.
It's also good to go to students too.
No
Like
offense, but professors sometimes are like, oh, this is exactly how I understand it,
Yeah.
so this is how you should understand it, but like getting a different background.
Are you ever like available for small tutor sessions at
Yeah.
all? Okay, I wasn't sure like. If
Yeah,
you
no,
were
you
open
can
to
just
that.
email me.
What's your last name? Okay. Cause I know like, I obviously go to like tutor sessions within the class, but you know, you've done this class already and succeeded, so we're all going through it together, so. Yes.
And then your last name.
Cox, C-O-X.
S.
Yep, S, that is me.
I will send you an email from email.
Cool, so I'll have it.
Yeah, so you can definitely shoot me an email. I check it probably every day, at least twice a day.
Okay.
Weird.
No, I check mine like 17 times a day, so as if there's something new gonna be in there.
I'm like, oh, the bookstore's having a sale.
Right, right, oh, random
They
stuff.
just send me a lot of things.
Yeah, yeah,
Okay, I just sent it.
okay, sounds good, thank you guys. Have a good
Awesome.
night.
See you next
awesome.
week.
See you. Yeah, that'll be good. Thank you. All right. So I think somebody asked about haplodiploidy. So here's a big mess without the image. So I'll be in cyan for the context here. So the big thing is that male bees are haploid. They only have 1 copy of each chromosome. That means no crossing over, no assortment, no variation. The same contribution is going to be given to both sisters in this case exactly. So off the bat, they're 50% the same exact thing. Then the mom, the queen, is going to give a variable gimme. These can cross exchange, crossing over, blah, blah, blah. So you have a good, nice mix and a randomized gimme. That means that if you're already starting at 25 or starting at 50% already alike, the remaining 50% comes from mom. 50% divided by 2, and that's the possibility that you're getting the exact same material, is only 25%. Hence, 75 total percent. So add on the 50% clone from your dad as a B, and then the mixed genetic blueprint from mom, meaning that these two sisters are 75% alike, mainly attributing to their drone father in this case. Hopefully that's okay. Do you all have, yeah, if you guys had any other questions, I'm happy to stay for a couple.
I think that helps.
Okay.
Thank you.
Yeah, insects are weird, sorry. But it's a good example of principle, so. Cool. I'm gonna turn these down.
Dr. Harder, I just have a question on kind of like format of class.
Yeah.
So I saw in the grades that we have 2 midterm exams. So is the first one at like actual midterms?
Oh,
or-
no, they split the difference, sorry.
Oh, okay.
So yeah, just check out the syllabus and the dates. They all should be in there. I'm not 100% sure when, you know, quote unquote midterms technically are. I kind of just designed it so that coming back from spring break, it's not like a week later or something.
Oh, okay,
Because
perfect.
I figured that's kind of terrible. So
Yeah.
Did I hear you say that the exams are open note?
I think that's probably the only way we're going to be able to do it.
Okay,
I'm still
but
kind of
they're in person, aren't they?
yeah, about that. I don't have a big enough room, probably.
Okay.
So even our massive room that fits 45, 50 people. There's 2 sections of us. So what would have to happen then is like, yeah, maybe 50% of you get to take the exam in person, but then I have to schedule the rest of you somehow in person. And I don't really think that's going to be cool.
Okay.
That being said, maybe I'll pull some strings. I don't know, to be evil. We'll see. But I think actually another option that we might do is if you want to take the exam in person and with your notes, and I'm physically there to help, that's cool. And then during the same time, there'd be an online version that, again, you have all your open notes and stuff. I think the only stipulation that I'm trying to go for is, you know, don't take the test literally with some